Today we will discuss about the effect of TGF-beta on glycosylation. So far, if you are following this channel or if you are following this biology about this TGF-beta, you might aware that TGF-beta generally affect this EMT, that is epithelial to mesenchymal transition. Epithelial means cells remains at their state, and whereas mesenchymal in common language means that it has the ability to move from one place to another place. So it's the TGF-beta that plays a major role towards acquiring the cancer cell its metastatic state. Along with TGF-beta, there are other factors also, but TGF-beta cytokines also plays a role in that, in most of the cancers. Now we will come on our topic, that is effect of TGF-beta on glycosylation. So in the normal conditions, cells attach to each other, and this will also helps the cells to remain on its place. And at this is the conditions which is commonly called as epithelial conditions, and we can find the epithelial markers are high in these conditions. And some of the epithelial markers are mentioned here, that is E-cadherin, β-catenin, claudin 1, Occludin. And the glycosylation structures are also well defined for the mammalian cells in the normal state. That is, it contains the hybrid structures, hybrid N-glycans, O-glycans such as GG4, GM2, and this sialyl, this O-glycans with this N-acetyl-galactosamine and this oligomannose. And usually what happens in the N-glycan in the normal conditions, the core structures remains constant. That is, this 2 N-acetyl-glycosamine and this 3 mannose, it remains constant, and above that this composition changes. So this in this case it is the oligomannose. It has the 3 mannose apart from the core structures of the N-glycan. So, and this is in the epithelial conditions. In the mesenchymal conditions, the glycan structures varies, and at the same time the cell-cell interactions weakens, and that will also facilitates the cells to move from one place to another place. So what are the changes that commonly found in the mesenchymal type cells is that it will have the glycan structures which has more branching, that can be seen here, and it will also have this alpha-2,6-sialic acid. So sometimes this is also used as a marker to find out whether the cells are in mesenchymal state or not. At the same time, the O-glycan structures are also changes, and the commonly found O-glycan structures in mesenchymal markers are STN and TN antigen that are not shown here. Here only this N-acetyl-galactosamine with galactose shown here, but if it is attached with the sialic acid, then and with some modification, and it is called this TN and STN structures, and we have discussed those. If you want to know the exact structures of this TN and STN, you can refer to our previous videos about the glycosylation in cancers. So you will find the link in the I button. And apart from that, the O-glycan structures from this GG4 and GM2, this gets diminished, and this GM3 is usually overexpressed. And this Levis A, sialyl Levis A and sialyl Levis B structures are also commonly expressed, along with the this sialations. Over sialations are also found in the N-glycan structures compared to the normal case. So these are some of the changes that occurs, and sometimes these changes is also attributed to the cancer state, which completely makes sense, as the cancer states which acquire these metastatic conditions usually have the change in the glycan structures, and most of them are found to be different from the normal structures. And these are some of the common glycans that are found in most of the cancers. And some of the mesenchymal markers are also shown here. So in these conditions, the epithelial marker goes down, and if one checks these mesenchymal markers, it might have gone up. And one of the common strategies that people used is qPCR. that's the RT-qPCR, we can say that, real-time quantitative PCR. And some of the measurement markers that are shown here are, and common measurement markers are N-cadherin, Vimentin, fibronectin, snail 1-2. So this is how in general the TGF-beta cause change in the glycosylation from epithelial to mesenchymal. So I hope you understood the basic difference between the glycosylations in two different conditions. If you want to understand more about this topic in detail, you can go through the review paper. This link has been provided in the description. So that's all for this video. Thank you for your time.